Chapter 6 and 7 focus on DNA, gene expression, and biotechnology. So in previous chapters, we've looked at where DNA is at. We've looked at what DNA is in general, what is the molecule? What is it used for? And so what we're going to be focusing on this chapter is it's going to be a little bit of review about what DNA is and what it's used for, but mostly focusing on how is it used in the cell? So that's the gene expression part. And then biotechnology, how are we using that information to work on solutions for humans as far as our own health and as far as things like crops that we grow. So that's what chapters 6 and 7 are going to focus on. And the thing is that we already know a little bit about DNA, like I said, from previous chapters. So in chapter 3, we talked about DNA as one of the macromolecules and what it does. So DNA, what is it? It's one of the macromolecules. And I've got some questions further on to do a little bit of review on it. What does it do? It basically is the guide for how to build organisms. And you are living in incredibly exciting times right now because this is the era of biology. So you may look back in history and see that there was like the era of physics and the era of chemistry, all the other major sciences had kind of their day in the light. Well, now you are living in the era of biology. And right now with what you've seen going on in past with COVID and so forth, you may be able to get a little bit of an idea of what that means. Right? I don't know how many of you keep up with the news. You can see here some different headlines. You'll come across this kind of stuff all the time. New research coming out about different things like stem cells. Now you've probably heard a lot about mRNA and so forth. So, how do our genes code for things like our personality and diseases and things like that? And so that's what a major part of research is in the field of biology right now. So DNA, it is a molecule that all living organisms carry in almost every cell in their body. There are exceptions. I think I mentioned in a previous lecture, for example, in humans, we have DNA in all of the cells of our body except for our red blood cells, since their job is to carry oxygen. It makes sense, hopefully, that they don't need DNA. They also get recycled on a very regular basis, so they don't need the directions, but the stem cells that make them in your blood marrow do have DNA coded on how to make those red blood cells. So what does DNA contain? So what is the purpose of DNA? What is in the DNA itself? Yeah, hopefully you said something along the lines of the information on how to build an organism, right? And that is one of the two important features of DNA. So DNA contains those instructions on how to create a body and control the growth and development of that organism, whether it's a human or a plant or a fungi or a bacteria. It doesn't matter what the organism is. That's what DNA is going to do. It has those directions, those instructions on how to do that. And then the other important feature on DNA is not just that it contains the instructions, but those instructions are encoded and passed down from parent to offspring. So the way the DNA is constructed makes it stable and copyable so that those directions can be made into copies, and those copies can be passed from parent to child. Right? Again, whether you are a single cell like a bacteria or an amoeba, or you're a multicellular organism like ourselves, or a big plant like a tree, you're still going to be able to pass those directions down. So I do want to tell you a little bit on the history of DNA. There were a lot of different scientists who were involved in the discovery of DNA as far as its structure goes, and we've seen the structure before. So here is the original model that was actually produced and was involved in the Nobel Prize in Chemistry. And actually, I'm sorry, I think, this is the Nobel Prize for Biology. There were scientists involved in this that won Nobel Prizes for chemistry. So the two gentlemen that you see here, I'm not going to ask you to remember their names, but you may have heard of these two. This is Francis Crick and James Watson. I don't remember which one's which in the picture, but these two scientists are credited with figuring out the structure of DNA. What they did is they actually didn't do any of the work as far as the bits and pieces to put the structure together. They used other scientists' work to build the structure. So they used work from an American scientist called Linus Pauling, who, he was working on the structure. They also used a couple of English scientists who actually constructed parts of DNA, as far as their structure of the structure for sizing and so forth. So they were using other scientists' measurements and information to build this final model. And their model hasn't really changed since they presented this model back in the early 1900s. So this is the model of DNA. I mean, the sketches, and even if you look back here at this wire structure, it's pretty much stayed the same because we've been able to build the pieces and parts. There were scientists that also worked on figuring out the A, T, C, and G chemical part. So let's review a little bit on what we already know on on DNA. So I mentioned just a little bit ago, DNA is one of the macromolecules. Which one of the groups of macromolecules does DNA belong to? So remember, there's 4 macromolecule groups. There were the carbohydrates, the nucleic acids, the proteins, and the lipids, right? So those are the 4 groups. So which one of those macromolecule groups does DNA belong to? And yep, it's a nucleic acid. And then it's built out of individual units called nucleotides. You might not remember that one from way back, but it's got these nucleotides, which I've got spelled out on the next slide. So there you go. So it's a nucleic acid, and it's built out of nucleotides. If you remember from the nucleic acids, DNA is one, RNA is the other, and we're going to see RNA again in this chapter because both of them are used in what we call gene expression. This is how we read DNA and build things. So we've seen this before, but just a little bit of review. We've got those nucleotides in their pairs, and then we've got those nucleotides made up into that ladder type structure that twists, creating that double helix. And so this right here, this is the structure that Francis and Crick came up with from those other scientists' research. So the strings of nucleotides, the spacing between them, even the spacing on the double helix, because if you notice, the double helix isn't consistent twist. It changes on the size of the space between the twists. All right, so just a reminder too, because this will be important as we go through parts of this chapter. So we've got those sugars, phosphates, and bases that make up the nucleotides. The bases are the part that we're most focused on. So A, T, C, and G. How do these guys pair up again? Remember, apples in the tree, cars in the garage, right? So here is just a little refresher question for you. So which of these will base pair with this DNA sequence? So if this is one side of the DNA helix, what would the other side say? All right. And then if you'd like, here's another one that you can try. And if you're not sure if you've got the answer correct, you can always shoot me an email and I can give you the answer for that one. All right. So, like I said, DNA has the directions for building pretty much everything on Earth. We haven't really found any organisms yet that don't use DNA as their building instructions. Now, when we talk about microbes, we'll talk about viruses. And viruses are weird because some of them can use RNA, but viruses, as we'll talk about in that chapter on microbes, aren't considered living organisms either. So you can see though here, no viruses. We've got an amoeba that's eating a little bacteria. It looks like humans, we've got plants, insects, amphibians. All of the organisms that you are familiar with are going to use DNA to provide their instructions. Now, there are a couple of terms to talk about before we get into the nitty-gritty type stuff about DNA. So one is genome. So a genome is the complete set of a DNA for an organism. So you can say, you know, we talk about the human genome. So the human genome is describing the set of genetic instructions, the entire set of genetic instructions of humans. And you can find the genome in every cell of that organism with a few exceptions. For example, in humans, you wouldn't be able to find our genome in red blood cells because they don't have DNA. But we can take just a couple of, skin cells, or if you have seen the DNA tests that they do, where you can send your DNA off and they can, do some reading on it. What they're doing is they're collecting the skin cells from inside of your mouth that basically collect in your spit, and they're taking those skin cells and pulling the genome out of those cells, because each one of your nuclei has the entire genome packed inside of it. Okay? So in eukaryotes, you can find the genome, the entire set of DNA for an organism, in the nucleus. You cannot find the entire genome for a prokaryote in the nucleus because they don't have a nucleus. So the genome for a prokaryote is just going to be inside of the cell. Okay? Now that genome gets cut up into chromosomes. So you can see chromosomes are just one section out of the genome. And so they're just one or more pieces of DNA. So each organism is going to cut their genome up into different pieces. For example, you can see here, humans have 23 unique chromosomes, meaning that we've taken our genome and cut it into 23 chunks. So we could have the genome in one big chromosome, but instead we've cut it into 23 chunks. I like to think of it like 23 chapters in a book. And we also get 2 copies of each. So we get 1 copy from mom, 1 copy from dad. So we have a total of 24 of these chromosomes in each of our cells, if everything's gone correctly. And so in humans, we've got 23 unique chromosomes. So 23 copies, or unique ones that are different from each other, but we have 46 total because we have 2 copies of each. That's why I like to think of them as chapters, because we've got 2 sets of 2 books, 1 book from mom, 1 book from dad, 23 chapters in one book, 23 chapters in the other book. Okay? And you can see there that the chromosomes vary in length. We'll see that in future chapters when we see things like karyotypes, where all the chromosomes are laid out. And they can have different numbers of base pairs. So the base pairs are just those A's, T's, C's, and G's paired together. That's what we mean by base pairs. Now, inside of those chromosomes, you're going to find genes, and almost all of the chromosomes have multiple genes. And we've talked about genes in a previous lecture. Genes are specific sets of DNA bases. So you can see here, average of about 3,000 bases long. These are sections or sequences of DNA that contain the actual directions for making things like a protein. Okay, so we talk about our genome having all of our information, but the genes are the specific directions on how to make different things like proteins. And so we're going to read a gene, and that's what a lot of this chapter is going to be looking at, is looking at reading these genes or what goes wrong with these genes to make mistakes. But we read the gene on the chromosome inside the genome, right? But the gene is the smallest section that we're going to be looking at, and that gene has the directions on how to make a protein. So we'll pick up in the next section talking more about this idea of chromosomes and genes and what exactly makes up all of our DNA, because it turns out not all of our DNA is these genes.